This is the final part of olfaction and the final part of the chemical senses. We're going to bring olfaction and taste together so that we can experience flavor after discussing higher order processing in olfaction. So as usual, we want to talk about the higher order olfactory processing, or what's going on when we get to the cortex. And I'm going to start by this talking about olfaction in the environment, and I'm going to talk again about this idea of how do we perceptually group specific molecules together for smelling a particular odorant. How do we segregate all the molecules into their various components to go with the correct odorant. And it seems particularly complicated for olfaction, especially we rarely smell one chemical in isolation. So he gives us the example of coffee, which has more than 100 different molecules. Plus, a lot of times when we're smelling coffee, we can smell coffee even when other odorants are present, even when other smells are around. So if I come downstairs to the kitchen and there's orange juice there and bacon and the smell of coffee, I'm smelling all of those different smells, and yet it doesn't, all of those molecules are in the air, and yet instead of smelling some weird bacony coffee thing or something where those molecules get segregated differently, I'm smelling coffee at times, and then other times I'll be smelling bacon will seem like the stronger smell, the smell that I'm paying attention to. And how do we do this? How do we perform this kind of perceptual organization for olfaction? And you can see my green questions here. And then I also remind you of vision. If you remember talking about, we did this for vision, and we had a whole practice in class discussing this for hearing, how we might use things like similarity. I'll use the hearing of similarity of timbre and pitch. We're going to group that together into a melody, or group that together in maybe into a particular instrument, as opposed to if they differ in timbre and pitch, we're going to segregate those into different instruments. We might use good continuation, proximity, common fate, synchrony, meaningfulness, familiarity. So those are just some reminders of what we've talked about for other sensory modalities. And then I ask, what do you think? Anything else? What are we doing for olfaction? Again, I'm not smelling a bunch of molecules from the coffee as mixing with some of the molecules from the bacon. And vice versa, and other molecules mixing with different molecules, such that I go, what is that smell, right? Somehow I'm smelling coffee and bacon. I keep leaving out the orange juice because it seems like a smell that travels less, but if I put my nose up to the orange juice, I can smell the orange juice as well. And I do want to pause here. I do want you to think about that. I want you to pause this and think about that before I give some answers that students have given in the past. There is no, here's the simple answer. I'm gonna say I think that a lot more of this is top-down processing than what we've seen before. That the bottom-up processing doesn't really explain things for me, although there might be a kind of proximity if there is some segregation to where those molecules are in the air, and that they are closer to each other when they are from all the molecules from coffee, let's say, are going to be closer to each other or closer to the coffee, and that maybe we can walk through bits and pieces where we have some bottom-up processing, but to a great extent this seems to be we need to have the experience of smelling a particular smell before we can really segregate that from the other smells that are around it. And so I'm going to say top-down processing, but I don't think that's the whole answer. And I don't think there's an easy answer. I don't know how much people have examined this the way they have examined vision and hearing. So it's a fun thing to think about. So I have this picture again of the olfactory mucosa, that's where our olfactory receptor neurons are, the sensory neurons. and that those sensory receptors are communicating to the olfactory bulb, which is immediately communicating to primary olfactory receiving area, the piriform cortex, and the amygdala. There's lots of crosstalk here between those areas and the olfactory bulb. There's crosstalk here between those areas and each other, and there's crosstalk between those areas and the orbitofrontal cortex. And when we're going to talk about now higher order olfactory processing, remember this is getting information to the cortex, and really we're going to include the piriform cortex here, but I'm going to get much more specific about what's happening with that orbitofrontal cortex. And you can tell from the picture of the brain there. So this is named after, we have the prefrontal, it's part of the prefrontal cortex, and we have that kind of ventromedial prefrontal cortex in the middle there, and the orbitofrontal cortex is really kind of around the orbits of the eyes, if that helps you to remember that. It's just kind of right up above the orbits of the eyes, thus its name, the orbitofrontal cortex. So in the piriform cortex, odorants cause widespread activity here that can change via experience. If we go back to the olfactory bulb, that is a chemotopic map that is based on the chemicals themselves, the chain length, and our perception, what that odorant is going to smell like. And that's not going to change no matter how much we smell that smell, or whether it's the first time we've ever smelled that smell. That activity of the olfactory bulb is going to be the activity, just like visual information is going to be the visual information that we experience. But as we get to the piriform cortex, this idea that odorants cause widespread activity, that is what I was explaining before with the picture where we went into the piriform cortex, and I mentioned that it's not just communicating to the amygdala and the orbitofrontal cortex, it is communicating across the hemisphere to the entorhinal cortex, to the perirhinal cortex, to the limbic system, to so the amygdala and the orbitofrontal cortex as well. So all across that hemisphere, those neurons are communicating to many different areas, to thousands of other neurons, and that's widespread activity. And that activity is going to change based on experience. Goldstein gives us this nice picture of, let's say, so I have this odor object like a rose. I smell it. Every time I smell it, I'm going to have that same activity on the olfactory bulb, as this is just some example of where the olfactory bulb might be active. And then when I go to the piriform cortex, I'm going to see this scattered activation. Then as I smell that object, that odor object, more and more, we see the connections between the neurons are going to, the pattern of activation can change and the connections between the neurons can change. And this is just showing within the piriform cortex, let's say, those connections that are built based on my experience, but that's going to be connections, again, throughout the hemisphere. And so to some extent, this is going to be how it is going to change based on just how well do I know the odor object. So I usually do a smell lab or a smell demonstration, and one of those odor objects is coffee, and most students recognize that very easily. We've had lots of experience with coffee. That's going to change that activity in the piriform cortex, that you're getting more and more experience with this. But there's also going to be changes depending on your specific experience. So did you have a really good experience with smelling coffee? Have you had a really bad experience with smelling coffee? That's going to change those connections as well. And this Donald Wilson study, given time, we can discriminate between odorants. He did a study with rats where they gave them either isoamyl acetate, if I'm remembering that correctly, it's a banana type odor, and they gave them another odor object of the isoamyl acetate mixed with another smell. I want to say pineapple, but I think I'm mixing, a peppermint smell. And the rats were either allowed to sniff the odor object for 10 seconds, so about 20 sniffs, or they were allowed to sniff those odor objects for about 50 seconds, so about 100 sniffs. And if we look at what's happening in with their experience and with the pattern of activation for those odor objects in the piriform cortex, later on we did not see much difference between the two smells for the conditions where the rats could only smell that for about 10 seconds, as they were not getting enough experience to really distinguish those. But if they were given 50 seconds to smell each of those smells, they were able to distinguish those. There was a difference for them in their perception of the two odor objects, and they could tell the difference. And we see a change in the pattern of activation in the piriform cortex. When we get out to the orbitofrontal cortex, that is now assigning an affective value to stimuli and experiences. And so that's, if you remember, that communication is going both ways, and it's talking back, communicating back to the amygdala and to the piriform cortex, which is why I said whether or not we've had good or bad experiences can influence that activity in the piriform cortex, as that orbitofrontal cortex assigns an affective value. In this study, they gave people, this is a little bit gross if you think about it, but they gave people a mixture of isovaleric acid. So this is something that smells a bit like sweat, and a cheddar cheese flavoring. And if they were told this is cheddar cheese, people rated that as more pleasant than if they were told this is body odor. They rated that as less pleasant. Again, it just doesn't sound very pleasant at all. But apparently, if we're told, if it has that cheddar cheese flavoring as part of this, as part of the odor, and we're told it's cheddar cheese, maybe we don't notice the isovaleric acid, this kind of smell, sweat smell, so much. And then if we're told it's body odor, obviously we do, and that cheddar cheese flavoring is probably not a great smell to have in there, and so rated as much less pleasant. And we see differences in the orbitofrontal cortex. I think I actually have this on a slide here. I'm going to go ahead and move while I'm talking this time. Yes, where if we look at fMRI activation, this is a percent change, right? They have a control condition that they subtract out of this, out of examining the cheddar cheese or the body odor. And you can see that if we compare this to a control condition, and I'm told that it's cheddar cheese, I'm rating this as pleasant, and I have activation in the orbitofrontal cortex, okay, excitatory activation in the orbitofrontal cortex. If I'm told this is body odor, I not only rate this as unpleasant, but it almost looks like I'm inhibiting activity in my orbitofrontal cortex, as we are below my control condition in this case. I think that's interesting. We're actually just kind of doing this, don't like it, and we're inhibiting activity there. And this is the place where we're going to talk now about flavor, as we're going to talk about it as occurring in this orbitofrontal cortex, that we're putting all this information together so that we can experience flavor. But first we're going to see how it gets there. Okay, so flavor is taste plus olfaction. And in fact, you'll notice that if you ever have had a cold where your nose is really stuffed up, or you've ever just pinched your nose in order to take medicine, that when you take that olfaction out of the equation, that there's very little to taste. And so I'm often asked questions about spicy foods, and not just the spicy foods that actually influence the chemoreceptors in our mouths, like capsaicin, like hot pepper, but also just Indian foods and curry and things like that. So there might be some small components of taste that I'm not exactly sure whether we're tasting very much sour or bitter, or little bits of taste, but really where we're getting that experience of something like eating curry, a great deal of that is happening because of olfaction. And when I tell you to do the demo there on page 378, that is exactly just eating a couple things, or drinking coffee or something, while pinching your nose and noting the lack of flavor, and then taking your hands off your nose and then noticing how it just wafts into your olfactory mucosa, and it's a much richer experience. It's a completely different experience. And what's happening there is when we're putting that food in our mouths, some of those molecules are going up through this retronasal route, through the nasal pharynx, as it's called, and that's going up and hitting those odorants are going up and hitting the olfactory mucosa. So flavor is going to put together all of this information. And your author includes this data now looking at particular participants and how they rated 3 different flavors. One is sodium oleate, which has a kind of soapy taste, And you can see that when the nose was open, so they were not clamping their nose, that most people rated it as soapy. A few people rated it as something else. And then they had a condition where they clamped the nose shut, and most people rated this now as tasteless. We see something similar for ferrous sulfate. When the nose is left open, most people rate that as kind of metallic tasting. When the nose is clamped, it gets a different taste, and a lot of people rate it as tasteless. MSG, or monosodium glutamate, which we have talked about as the example of a salty, chemical. This has really has the same flavor whether the nose is clamped or not. I actually talked about it as a bit umami, but people rate it as kind of salty, and they rate it as kind of salty whether they have their nose open or clamped. So that's one of those examples of something that we're really experiencing the taste as the flavor. But usually what we do is add MSG to other food. So many of the sensations that we call taste, and I make this mistake a lot of just calling something taste when what I really mean is flavor. Much of what we call taste is actually stimulation of the olfactory receptors, and some are more influenced by the taste receptors themselves, and some are not so influenced by the taste receptors themselves. They really require olfactory receptor activation. As I was just talking about, curry is a decent example of, if you pinch your nose when you eat a little bit of curry, it really doesn't seem to have much flavor at all. There's not a lot of the taste cells as a component of that experience. It's really greatly influenced by olfaction. So a number of cortical areas that serve both taste and olfaction are probably involved in our physiology of flavor and how we experience flavor. But most of the research has been done on the orbitofrontal cortex, where we really see all of these different sensory modalities coming together so that we have this experience of not just flavor, but of our food, of the experience of eating our food. This is where taste and smell responses are first combined into something more complicated like flavor. But the orbitofrontal cortex also receives input from the primary somatosensory cortex, so the feel of our food, and the infratemporal cortex, so the looks of our food. The infratemporal cortex in that visual what pathway, so where we do our object recognition. If you've ever done the green eggs and ham when you were a little kid, my sister does that with her 4-year-olds, usually every year. Guess they're probably not doing that this year, but and as the kids are rating, she also collects descriptive data and shows them their descriptive data of who liked the green eggs and ham versus not, compared to regular eggs and ham. Some children don't like the green eggs and ham, even though it's exactly the same as the regular eggs and ham that they eat. That information, those eggs being green and that ham being green, just that influences their experience of the food. And certainly how food feels in our mouth is very important as well. But the orbitofrontal cortex does not just receive input from the primary cortical areas for taste and smell, so the insula, the frontal operculum, the piriform cortex, the somatosensory cortex of the feeling of our body, of our mouths in this case, and the infratemporal cortex, so what that food looks like. But I have had students do their project on the importance of the sound of the food, so the sound of popcorn popping, or the sound of our potato chips crunching. That also influences our experience of food. And the orbitofrontal cortex is putting all of that information together for flavor with taste and smell, but also for just the overall experience. I think he talks in the book a little bit about, I don't know if he took this out, about a particular soup that he has eaten that has the hints of lemongrass and the depth of mushrooms, and he has all of these flavors that are combining to make this really amazing experience of a soup. And the orbitofrontal cortex is doing this via bimodal neurons, so neurons that respond to more than one sense. So far, we've mostly talked about neurons that are responding to vision or neurons that are responding to hearing, and we have to put that information together. But we do have bimodal neurons, and they tend to respond to similar qualities in the environment, or qualities that are going to tend to occur together in the environment. So for example, a particular neuron that responds to the taste of sweet fruits would also respond to the smell of sweet fruits. And the orbitofrontal cortex is said to be the cortical center for detecting flavor and the perceptual representation of foods, as we are now experiencing several modalities for our experience of food. And we're putting them together with these bimodal neurons. I think your author has a couple of examples of studies conducted on the orbitofrontal cortex, but I am running long, so I'm going to just do this one, and we're going to see something really similar to what we just saw in the orbitofrontal cortex for that olfaction study, where they looked at a pleasant smell, what people rated as pleasant, so cheddar cheese, versus unpleasant, the smell of body odor, even though, again, the odors themselves were the same. They were created in the same way. It's just what is my effective labeling of that was influencing the orbitofrontal cortex. And they also did this with ratings. So they gave people a number of wines. If I remember, there were 5 wines, but actually there were only 3. So they're told, the participants are told that there are 5 wines, but 2 of those wines are the same, and then another 2 are the same, and then there's one that's different. And what they do is they give them, so if I'm going to use the wines examples A and B. I have the wines A and B. They're actually the same wine, but they tell me that A is a wine that costs $10 and B is a wine that costs $90. Okay, and so what is my enjoyment of those different wines? Even though, again, they're the same wine, the only thing that's different is what I'm told about the price of those wines. And then they have another condition where they participants don't hear anything about the price of the wines. And as you can see, if there's no price given, people are rating them the same pleasantness. They're actually the same wine, as we're putting some of those wines together in the same category. But then looking across those wines that are exactly the same, if that makes sense, this is always a hard study for me to describe and explain, but so if I'm told that the wine was a $10 wine, I rate it as less pleasant than if I'm told that that wine is a $90 bottle of wine. I rate that as more pleasant. Although, again, this is the same wines in these 2 conditions of $10 or $90. As we see this change in ratings, just as we saw before with the affective component of that smell, we also see similar activation in the orbitofrontal cortex, or activation that matches really my pleasantness rating. So if I'm told that it's a $10 bottle of wine, you can see this again in the blue line, again, almost this inhibition, what looks like inhibition of the orbitofrontal cortex, as I'm not enjoying that wine. And then as if I'm told, same wine, that this is a $90 bottle of wine, then I'm not only rating that as that I enjoy it more, but I see that activation of that excitatory activation in the orbitofrontal cortex, that I'm really labeling that with something positive. So I'm going to return one more time to this notion that this idea of how smell goes in from those sensory receptor neurons to the olfactory bulb, no thalamus, and then goes straight to the amygdala and the piriform cortex, and that piriform cortex communicating with those really emotional areas, those limbic system areas, and those emotionally associated memory areas like the amygdala, but also our memory areas like the pararhinal cortex, the entorhinal cortex, and the hippocampus, that these odorants, these smells, have a really strong influence on our emotion and that feeling of having a more vivid kind of memory. This is often called the Proust effect, as Marcel Proust wrote about this very eloquently back in the early 1900s in The Remembrance of Things Past. He's describing this experience with these madeleines, this particular cookie, and he says, the vision, the looks of them don't really bring back any specific memory, but as soon as he starts eating them, of course, the flavor, the olfaction, all of that whole experience brings him back, transports him back to being a child and the house he lived in and the square on which he lived. He has all this just rush of memory. If you've ever met somebody who has, or if you've ever gone back to someplace where you lived when you were a young child, so we moved around a lot, and sometimes we go back to Huntington, West Virginia, it brings back certain memories, and some of that, of course, is the sounds that are around or the particular sights that are around, but a stronger effect really is the odorants, the smells that are in the air. As we see, this olfactory-induced memories have this emotionality, and they have this feeling of being really vivid. They aren't necessarily more accurate. So just like those memories that are of kind of traumatic events, if you've seen this research before, where we see that the amygdala is involved in those memories, while they feel more vivid, they feel more accurate. When they've done the research, it looks like they are not necessarily any more accurate. They just have more kind of emotionality to them. But the way we bring up memory, and then we talk about it and think about it, and then we lay that memory back down, we reconsolidate, and it is biologically changed. And that's just as true for these kinds of memories as other kinds of memories. I'm going to, one more time here, suggest that you do that, chopping up some fruits or finding some spices and giving them to a friend to smell, and having your friend do the same thing for you. Just 2 or 3 or 4 of those to get this experience of, and taking out the vision, getting this experience of, can I put a name or label on this smell, but also noting, does it remind me of something? Does it bring me back to a particular time? When I've done this kind of demonstration in class, it doesn't work every semester, but many semesters I've had students, especially for those that kind of category of spices like nutmeg and cloves and cinnamon, they talk about being reminded of their mother's kitchen or their grandmother's kitchen, or aspects of the holidays. And one semester I had this really neat experience where I had a Middle Eastern woman, so a woman from one of the Middle Eastern countries, in my class. I was using garam masala, which is one I often use, as it's kind of a mixture of spices, and students usually make guesses that are some of the spices that are in it, but most people aren't really aware of garam masala, so it's just kind of a fun one for me to make you all guess and think. But she had that experience of just really being reminded strongly of home, because in her country, every household created their own garam masala, kind of the way we do bog in the Low Country, where if you go from home to home, there are slight differences, and some people will go, well, I do my own thing, and I add this. They do that with garam masala. So it brought back a lot of memories for her, and that was I thought, a neat experience for everybody to kind of hear her talk about that. But I do recommend that you go have that experience in one way or another of just smelling some smells and seeing, again, both how well you name and label those smells and how difficult that sometimes is, as well as this, what are the memories that I have, or is this reminding me of a particular memory, and how vivid is that memory. Okay, I am running a teeny bit long, but not for the overall. This is just going to be a long video itself. Do read the eReserve journal article. I'm really excited about you all discussing either taste, or the lingual tactile sensitivity, or olfaction, which, again, that connectivity of piriform cortex is one of my favorite articles maybe ever. And you can see how often here I've gotten excited about exactly what you're going to read about there.